Welcome. It's a pleasure to welcome Bo Lee back to the Dana-Farber. Bo started out in physics at Peking University, and then did a PhD in bioinformatics at the University of Michigan with June Lee, and then came for a postdoc to Boston with Shirley Liu and June Liu at the Dana-Farber Cancer Institute. Among multiple honors he's received, he was a Benn Scholar while a postdoc. He's focused his work on T-cell receptor analysis and genomic-based cancer detection. And we look forward to hearing his information about that as he tells us about challenges and clinical opportunities in immune repertoire analysis. He's currently an assistant professor at the Department of Bioinformatics and Immunology at UT Southwestern Medical Center. And we look forward to his talk. Please post your questions in the Q&A section, not the chat section, and I'll relay them after his talk. Bo, it's yours.
Thank you, Gordon, for a great introduction. And thanks everyone for giving me the opportunity to present in this great seminar series in oncology at Dana-Farber. It feels really good to come back even virtually. Today, I'm going to talk about our recent research work on solving the challenges and exploring the clinical opportunities in computational immune repertoire analysis. I was motivated to step in the field of cancer immunology by the success of cancer immunotherapies. And everyone knows that there's a Nobel Prize in 2018 awarded to Dr. James Allison and Dr. Hongzhou. But really, I mean, there are great heroes in the field that have made the equal contribution to the field. I think all of their contributions are exceptional to make the clinical success of immunotherapy possible. And for myself, I think my early contact with Dr. Gordon Freeman and Dr. Arlene Sharp are basically one of the most biggest incentives for me to step into the field. But I was thinking about something different from therapeutic applications, because I'm not a doctor, so it's really hard for me to think from a clinical perspective. So what I can think about is how can we use immune receptors, for example, to detect cancer? The idea hit me in the morning of 2015 when I was walking the intersection of the Longwood Avenue and the Brookline Avenue. I think, I mean, for most of the audience here, you must be familiar with how busy it was in the morning, a normal workday morning. At every signal, there could be like hundreds of people crossing the intersection. And my knowledge told me that there are about 3% of the population that has cancer, either as diagnosed or probably unknown. So when I watched that crowd, I thought that there might be some cancer patients that they don't even know they have cancer at this time. So is there a way that we can do a fast and accurate and non-invasive cancer diagnosis? Perhaps since I'm doing immunology, is it possible that we can do that from the immune receptor angle? And of course, I think about T-cell receptors because it is most important, one of the most important cell type in the immune system. I think it is a good idea because the immune system reacts early thanks to immunosurveillance. I think for this audience, it is really not necessary for me to introduce what immunosurveillance is. And second, after antigen challenge or antigen stimulation, the T cells can proliferate themselves hundreds of thousands of times. So this significantly amplify the early stage cancer signals. And finally, it can be detected in the blood because these are the circulating T cells and can be assessed in a non-invasive way. But the challenge is also very obvious. A healthy adult can have hundreds of millions different T cells in the blood. Of these T cells, they are specific to, I think, numerous different kinds of antigens. And how can we figure out which of them are killing cancer? Back in the days of 2015, really little is known about cancer-associated T cells. So I spent many years in this direction to prepare for this task. We developed a series of tools to starting from calling the TCRs in the tumor samples so that we have a data set that could be enriched for cancer-associated T cells. And then we develop an algorithm to do clustering to prioritize the cancer-associated TCRs. And then we develop a deep neural network model to perform de novo prediction of these cancer-associated T cell receptors. And finally, we have another algorithm that help us to separate the TCRs from the to use TCRs to separate cancer from the other immunologically related diseases, such as autoimmune disorders or acute infections, etc. So all these tools took years to develop, but we have only one goal, and one goal only, which is to use TCR to diagnose cancer, and hopefully we can find it at early stage. So we begin from the first step. That was my postdoc work. I was at the Dana-Farber Cancer Institute from 2014 to 2017, and this was the most important work I developed back in the days at the Dana-Farber, which is called TRUST. This algorithm is able to identify the T cell receptor hypervariable sequences from the tumor genomics data, the bulk tumor RNA sequencing data. I think for people who are not very familiar with the T cell receptor, it is a heterodimer. It's a heterodimer consisting of alpha chain and a beta chain, and both alpha chain and beta chain has 3 CDR loops. CDR is short for complementarity determining region, and all 6 loops of all the 6 loops, the CDR3 regions are the most diversified region because they are generated from the somatic VDJ recombination process. This chart is for immunoglobulin heavy chain, but the TCR alpha and the beta chain basically follow the same logic. Basically, there are multiple variable gene and diversity gene and joining gene in the DNA sequence. And during the recombination, the cell will randomly pick a variable, a diversity, and a joining gene and put them together and forming this VDJ junction. The VDJ junction is super diversified because not only that the randomness of the picking of V and D and J, but also during the junction formation, there are random nucleotide insertion and deletion in the VD and the DJ junctions. So eventually the CDR3 regions have a huge diversity, theoretical diversity that basically make us a very versatile TCR repertoire that is able to recognize all kinds of different foreign and internal antigens. So we were genomics computational biologists, so we want to look at the TCGA data and try to figure out if it is possible to identify the DNA sequencing reads that is generated from the TCR transcripts. And if we align the we align the reads in the TCR variable gene region and according to the reference genome, and we find that these reads align perfectly well with the variable gene location, which means that we do identify the transcripts that are derived from the T cells in the tumor, which is good. That means that it is theoretically possible to capture the CDR3 sequences. And when we further zoom in to the end of the variable gene region, we find interesting repairs that is showing that one pair mate is perfectly mapped to the end of the variable gene, but its paired mate is unmappable, which is just as expected because these reads contains part of the hypervariable CDR3 region. There's no way that the aligner can find a region to map it to the reference genome. So it's discarded as unmappable. Therefore, we would be able to retrieve these very valuable treasured CDR3 reads out of the unmappable trash can. So that is our trust algorithm is about. Basically, we use the parent information to retrieve the unmappable reads based on the paired mate is mapping to the variable or the joining gene. And then we divide these mapped reads into, so we do a pairwise read comparison using Smith-Waterman alignment. And then we divide that into disjoint clicks. And for each click, we will assemble them into a new contig. And eventually that new contig is actually a CDR3 containing transcript. So from the TCGA data, we were able to call, I think, a little bit over 1 million such transcripts. And that huge number for the first time allowed us to do a statistical pair up between the recurrent CDR3 motifs from the T cell and the recurrent new antigens or mutations that come from the cancer patients. So we look at the pairs that co-occur more often than expected, and we were able to identify 3 significant pairs. Of them, this PRAMEF4 300V mutation significantly co-occur with this GESEQY CDR3 motif. And all the 3 patients that harbor this F300V mutation and the motif, they all have a HLA allele that is predicted to bind the mutated peptide, suggesting that these patients could really develop an immune response against this potential new antigen. But that was actually the early work when I was in Dana-Farber, and I can access limited clinical resources. So that is actually the end of the story, unfortunately. So after I became independent at Dana-Farber Cancer Institute, I really pursued a lot more downstream experimental and clinical follow-up. So the first task is really to from the large pool of the T cell receptor sequences we retrieved from the TCGA data, we want to prioritize which of them are truly cancer associated. And we want to increase our statistical power. As you can see, this is really hard because there are so many different neoantigens and so many different CDR3 motifs. This analysis is very preliminary. So we developed another algorithm, which is called iSmart, that is able to cluster similar CDR3 sequences together to gain statistical power. And that is really one of the necessary steps because of the diversified interaction between the T cell receptor and the tumor antigens. So this is already shown before that CDR3, the T cell receptor consists of alpha chain and the beta chain. And as I mentioned before, the TCR-CDR3 region is extremely diversified. So if you look at the middle part of these sequences, they are almost random. Of course, I mean, T cells know what their functions are, but by just eyeballing these sequences, I mean, we have no idea. But on the other hand, the counterpart, the tumor antigens, are also coming from extremely diversified resources, such as somatic mutations, structural variations, and so on. So there are multiple. I think the list is getting longer and longer. Eventually, we have limited knowledge about what kind of new things does the tumor produce, and which part of them can be recognized by the T cells. So, and to add another layer of complexity, it is currently impossible to computationally match the T cell receptors and to their cognate antigen targets. Not even with AlphaFold. I have tried that. No, it's not possible. Okay, so the state-of-the-art analysis of the TCR repertoire is through clustering of the similar TCR sequences into groups and assuming that the TCRs in the same group specific to the same antigens. So to make that argument simple, we use shared CDR3 motifs to serve as surrogates for shared antigen specificity. This notation or this notation was first brought up by 2 back-to-back Nature papers published in 2017. One is by Paul Thomas group from St. Jude Hospital, and another is by Mark Davis group from Stanford. So I'm using Mark Davis paper as an example. For example, they developed this glyph method to cluster CDR3 TCR sequences with similar motifs. And they use this influenza-specific T cell receptor as an example. So this particular peptide is a Nymer peptide that's derived from influenza flu M1 gene, and it's a immunodominant peptide. So in many individuals, we will find this the trace of infection because the repertoire, the T cell receptor specific to this antigen, they all carry this RSS motif in the middle. So I think you can appreciate this high similarity across multiple different T cell receptors. In the other words, if you find a TCR that has this specific length and a specific motif in the middle with RSS or RSA, you probably can make the assertion that this is an influenza M1 specific T cell receptor, even though you don't know actually. But when you see this motif, you know that this is something that is specific to a known antigen. But of course, when there is nothing is known, if you see 2 TCR receptors, they are very similar, you know that they could be recognized to the same antigen. All right, so we naturally wanted to use glyph to analyze the cancer genomics data. So we did a benchmark of this algorithm. We selected 2,000 T cell receptors from the public domain. They are specific to 15 different kind of epitopes. And most of them are viruses like HIV or dengue virus or EBV, etc. But but we applied glyph to cluster these 2,000 T cell receptors. The expectation is TCR specific to the same antigen will be clustered in the same group. But TCR specific to different antigens, on the other hand, should belong to different groups. But that is way not what we observed. So for example, we see a lot of TCR specific to KRW antigen, and this HIV epitope, and this RAK antigen. They are clustered to the same groups. And so these off-diagonal signals are all noise. They are all non-specific clustering. So you can see that glyph is not very specific. Actually, this is something a common knowledge in the field that you cannot really trust glyph to do this kind of task because it's not very specific. So that kind of motivated us to develop our own algorithm. We call it iSmart, which simply performs pairwise sequence alignment based on Blossom 6-2 protein substitution matrix. And it built connectivity matrix of the CDR3 sequences based on high alignment scores. And finally, it calls CDR3 clusters based on the connectivity matrix. So glyph is much more specific, as you can see. It's almost all the signals are along the diagonal, which means that the TCRs, only TCRs specific to the same antigens are clustered together. So we build this pipeline. We call the TCRs using trust from the TCGA data, and then we only select the complete ones, and then we excluded the TCRs that also seen in the healthy individuals because these are public TCRs, they're not cancer specific. And then finally, of the remaining, about half of the 80,000 non-public TCRs, we use iSmart to do clustering, and we got 4.6 thousand antigen specific CDR3 clusters. This is what the data look like. So if you, for example, if you take this one as example, you can see that the testicular germ cell cancer patient and the ovarian cancer patient share the T cell receptor. They're very similar. Even though the two patients never see each other before, somehow they have a very similar T cell receptor in their tumor. And the two sequences only differ by one amino acid, which is this tryptophan is replaced by a phenylalanine. And you know that these both are very large hydrophobic residuals. So it is highly likely that both of these are specific to the same antigens. So we take a thorough investigation of all the clusters we called, and we find some very interesting ones. For example, these two clusters are specific to, specifically enriched for colorectal cancer and endometrial cancer. By enriched, I mean the patients who has these T cell receptors are diagnosed with endometrial cancer or colorectal cancers. So we naturally want to explore why these T cell receptor or these patients are so different. So we compare the gene expression pattern of these patients, the patients with these TCRs against these without the TCRs, and to do a differential gene expression analysis. And we find that on the top of the list is this HSFX1 gene, which is short for heat shock protein transcription factor X-linked 1. It's a long name. It took me a while to memorize it, but I think it's a very informative name because it's X-linked and the transcription factor. Highly likely that it is tissue specific. And when we look across all the TCGA data of its gene expression, indeed, this is a very specific gene. So if you look at the expression pattern, it only expressed in colon, rectal, and endometrial cancers, as we expected. It is completely silenced in all the normal tissues, almost all the normal tissues, and the other cancer types as well. So it satisfies the definition of a cancer testis antigen. So the next step is to study if this is a true antigenic target. So we used, we predicted the HLA binding epitopes of this protein, HSFX1 protein, using the NetMGC online server. And we find this VMF dimer peptide to be a strong binder to 3 human HLA alleles, including HLA2, C701, and C702. And very interestingly, the HSFX1 expressing tumors all came from patients with a matched HLA allele. So this is independent information. Somehow every piece just matched together. So we performed this in vivo validation. We ordered the HLA-A2 humanized mouse from Jackson Lab, and we vaccinated the animal using the VMF peptide or negative control VRF. The replacement of this methionine into arginine completely abolishes binding to the HLA-A2 allele. So VRF peptide is served as a negative control. And we boost the animal using at day 14 using the same peptides, and then we collect the splenocytes and perform the interferon gamma-based ELISpot on day 18. And then you can appreciate that the treatment group have significantly higher count of spots compared to the control group. This is the control group. So from this experiment, we concluded that the VMF peptide is indeed a very strong binder to HLA-A2 allele, and the peptide itself can elicit a very strong T cell response in vivo. However, this is an animal experiment, and we know that a lot of human proteins can be considered as foreign antigens to mouse. So it is not surprising that there is a strong T cell response. So in order to really do a rigorous follow-up, we need to initiate the clinical trial. That is a little bit beyond our scope. So we did this kind of analysis. We ordered 20 high-grade endometrial adenocarcinoma samples from UT Southwestern data tissue bank. And we stained these tissue using the HSFX1 protein. And we find that the protein actually stains very well. It's very specific. And we find that 15% of the samples have a very strong staining only in the tumor region. While the adjacent normal endometrium is completely negative, suggesting that this protein of HSFX1 is indeed a tumor-specific biomarker. So if it's in the future, someone want to follow up with a cancer vaccine sort of therapy, this could be a very good target. And this is among the many similar predictions we made from our TCR clustering and large genomics data analysis. And we want to make the point that the T cell receptors we caught and clustered from the TCGA data are enriched for cancer-associated signals. So we want to use that as training data for our downstream algorithm development. Okay, so that is the third step, the third part of today's talk, which is, we want to develop a de novo predictor of cancer-associated TCR. That is a little bit tricky because I think it's a long shot. At that time, I really didn't know if this could work or not. So I was really surprised that it's working so well. So before I dive into the details, I'm going to introduce the concept of TCR repertoire. TCR repertoire is a collection of all the diversified T cell clonal types in the immune system. So blood plus lymphoid system, I think one healthy individual could possess over, I think 1 billion, probably 1 billion different T cell receptors. That's a huge number. And you could never imagine, what kind of T cells could recognize, what kind of antigens these T cells could pick up. But we need an extremely diversified repertoire to make sure that every possible antigenic encounter in our lifetime is taken care of. So that's why we need so many T cells. Okay, so the immune repertoire is generated from multiple steps. Of course, the first step is that the progenitor cells will generate a pre-selected pool of all the T cell receptors. And then they will undergo negative and positive thymus selection to give the naive repertoire. And during the lifetime of the environmental exposure, there are clonal selection process going on in the repertoire. So eventually, every individual's repertoire is different and their antigen experienced. So imagine the amount of information that is written in the T cell repertoire is super, super rich. So you have the lifetime infection or disease history encoded in the repertoire. If we have a smart algorithm that can help us to read that information, I think we will know a lot more than doctors. But unfortunately, at this step, that is actually far ahead. So we need to do a lot more to do that, to make it happen. But baby steps ahead. We first checked if the cancer patients, in the cancer patient blood, we can see more cancer-associated T cell receptors. That is a simple question, and it can be easily addressed. Okay, so we collected another 21 melanoma patient samples treated with CTLA-4, anti-CTLA-4 antibody. This is coming from another study published in 2014. This is not TCGA sample. And these are the blood samples sequenced by the adaptive biotechnology, which is the monopoly in the T cell repertoire sequencing field. There are 21 samples. They have both pre-treatment and post-treatment blood samples. We simply counted how many of the T cell receptors in these blood samples can be found in the TCGA data. And we use that count as a predictor. So you can clearly see that compared to the normal individuals, which come from a healthy control cohort, also sequenced by adaptive biotechnology, the same company, you can clearly see that the read count of the melanoma patients are significantly higher than the healthy individuals. This suggests that late-stage melanoma patients in the blood, there are a lot of circulating cancer-associated T cells. So if we use that count as a predictor, the AUC is like 80%. It's not perfect, but we see something. But this is not very interesting task because nobody needs to diagnose late-stage melanoma as using like a immunologically based approach. This is far beyond what is necessary. However, when we look at early-stage breast cancer patients, we see very similar trend that the breast cancer patients also have higher amount of cancer-associated T cells in their blood compared to the control. Although the signal is actually much weaker, and when you do a prediction, you barely see any prediction power. But this is proof of principle, right? It really proved the principle, right? It's possible that we can and we are able to. We already seen some signals in the blood samples of the cancer patients that there are some cancer-associated T cell receptors going on. So now we want to do a more sophisticated prediction. That is how we developed this machine learning algorithm. We use the TCGA data as the true positive cancer data, and we also collected a lot of TCR sequences from healthy individuals as true negative training data. Combining the two, we developed this two-layer convolutional neural network model. When we use the TCR data, we convert the TCR data as an input. We converted it as an image as an input, and then it's feed forward into two convolutional network layers, and then at the end we output a probability whether this T cell receptor is associated with cancer or not. So the model actually is not that complicated. The model only has about 3,000 parameters. Compared to the training data, this is actually very small. So we believe that the overfitting is not a big concern in this model structure. We purposely choose a small number of filters. We only have 8 filters for the first convolutional layer and 16 filters for the second. So it does not have a lot of parameters. Okay, but validation is very important. So we use the data set developed by Jenny Jiang's lab from UT Austin. They published a Nature Biotechnology paper in 2018. In this paper, they used DNA barcoded tetramers to sort the antigen-specific T cells in high throughput so that they can label the T cells with their cognate antigens with high confidence. And the antigen list include several neoantigens, cancer-associated antigens, oncogenic viruses, and non-oncogenic viruses such as influenza or yellow fever virus. So we just blindly applied DeepCat. By the way, this algorithm we call it DeepCat. So we just blindly applied DeepCat to predict the cancer probabilities for all the TCRs in these categories, and we compared them with control or non-cancer-associated TCRs. And we find that even neoantigen specific T cell receptors have some. We have some prediction power. By the way, this data set was completely unseen in the training data of DeepCat. This is completely new, and some of the neoantigen data, neoantigen specific T cell receptors is actually the new antigen targets was actually new in that paper. Actually, nobody has reported previously. So this is really an exciting kind of independent validation and motivated us to proceed to apply to more human cancer cohorts. So we applied to, I think this is about 15 or 17 different cohorts, including tumor infiltrating T cells, cancer PBMC samples, and non-cancer PBMC samples. All of them are TCR repertoire samples. Each repertoire will contain hundreds or even thousands of T cell receptors. What we do is we predict the cancer probability for each TCR, and then we average them out. We average them. We make an average to calculate the cancer score. So cancer score reflect the amount of cancer-associated T cell receptors in the blood repertoire, not necessarily blood. So when we look at the infiltrating T cell repertoire, we find that their cancer scores are much higher compared to the healthy individuals. This is the kind of a positive control here. So when we compare cancer PBMC and non-cancer PBMC, we can observe very strong upregulated cancer scores, compared to the healthy individuals, suggesting that it is in theory possible to use the cancer score as a new orthogonal predictor for cancer status. So that is the discovery cohort, and we also repeated our analysis using an independent cohort, which we spent 2 years time to collect. They're including mostly stage 1 or early stage cancer types. We have stage 1, early stage breast, kidney, ovarian, pancreatic, and lung cancer, compared to in another cohort of healthy donors. So all the samples are different. And now we see the same signal here, that even for early stage, even for stage 1 cancers, we can reproduce the signal that cancer scores are significantly higher in these cancer patients compared to the healthy individuals. So that is really exciting. But although exciting, there are some limitations of this approach. For example, it is still very preliminary because the sample size are very small. And second, when we apply the algorithm to study some of the autoimmune disorders, disease such as multiple sclerosis or arthritis patients, we also see some some elevation of the cancer scores. That is suggestive that the cancer score is not that cancer specific. It's more like a sort of autoimmune or tissue related kind of signal that is released into the blood. We are still trying to figure out the nature of that signal. Okay, so that comes to the last part of my talk, which is a new algorithm we developed last year, and that is able to perform ultra-fast TCR clustering. You may wonder what does that do with the cancer diagnosis. So please allow me to do a little bit digress, and I assure you that I will come back. Okay, so before we talk about clinical applications, now we ask a mathematical or computer science problem, a question: how to perform Smith-Waterman alignment on hundreds of millions of T cell receptors? This is pairwise alignment. So when you are performing a large number of alignment, using some sort of protein substitution matrix, the time complexity is quadratic, which is a nightmare. You begin with a small number, for example, thousands of T cell receptors, and your complexity is like you need to do millions of comparisons, which is okay. In Python, this is pretty fast. And TCR-dist, which is the state-of-art algorithm, can do it pretty well. And its sensitivity and specificity is very high. That is why TCR-dist is still one of the best algorithms in TCR clustering. When you scale it up to 1 million TCR sequences, you can still use TCR-dist, but you need to apply all kinds of computer science techniques, like a parallel, like you want to use probably GPU, you want to rewrite it into C++ and everything. You can make it to 1 million at affordable time. But when it goes to 100 millions, then forget about it. It will take you 5 to 10 years to finish up one batch. So that is impossible. You can't wait. So now it is not a computer science problem anymore. It is a mathematical problem. So how to do that really takes a new solution, completely different kind of thinking. So this is the solution we come up with. We want to define a numerical coordinate system that represents the similarity of the T cell receptors. So for example, we want to project the TCR sequence of SSGVTEA, this sequence, into a high dimensional space, for example, here. And a similar sequence will be projected in a similar location. And if this is possible, then clustering is simply the, okay, so TCR clustering problem is converted to finding nearest neighbors in a high dimensional Euclidean space. And that is log n times complexity. That is much, much faster because you can sequentially divide the space, like binarize the space. I mean, that is at log time scale, that is much faster. Okay, so this coordinate has to satisfy at least three qualities. First is what we call isometry, that the Euclidean distance has to be correlated with Smith-Waterman alignment score. And that is the most important property. And the second is non-commutability. That means when you switch the order of two amino acids in a sequence, your coordinate will be changed at least a lot. And finally, it has to be efficient, easy to calculate. And that is sort of why we are sticking to the Euclidean distance calculation. We do not want more sophisticated like geodesic distance calculation. And because that itself can take a long time to compute. And you cannot apply it to hundreds of millions of T-cell receptors eventually. So these three properties, okay, do they exist? We don't know. So we spent half a year of time getting the math framework ready. And finally, we were succeeded. So that is a good news. Okay, so the first step is to find a isometric embedding for the Blossom 6-2 matrix. Blossom 6-2 is the building block of our Smith-Waterman alignment algorithm. So you need that to calculate the alignment score of amino acid replacement. So we need to find a solution, isometric embedding solution for Blossom 6-2. Unfortunately, there is no exact solution because Blossom 6-2 cannot be written as a Euclidean distance matrix or EDM. It is there are two criteria to serve as the EDM. I think the first one is your dimensionality cannot be greater than 20. Here's 20 amino acid. And the second one has to be flat everywhere. And that is defined by some determinant. So you can check the original paper published in 1976. It does not even satisfy the triangular rule, like AB plus BC is bigger than AC. So it's not flat. Okay, but we can find an approximated solution by multiple dimensional scaling, MDS. You know, MDS is designed for this purpose, to find a approximate isometric embedding for any Euclidean distance-like matrix. It's not exactly the solution. So we produce 20 numeric vectors for each of the amino acid. We denote that by beta A. A is, for example, beta A is for amino acid alanine, and we have beta K for lysine. We have beta Y for tyrosine, right? Then we calculated the pairwise Euclidean distance, which is shown as the x-axis. We compare with the inverted Blossom score, which is 4 minus the original score, and it looks like this. So the correlation is pretty high. But it's not perfect. So there are some intrinsic noise that will be carried through the whole calculation. But the first step, I can tell you, this is the best we can achieve, and it's considered done. So the second step is really more challenging. It's to find a representation of the peptide sequence. How can we put together the peptide sequence and convert that into a numeric coordinate? So we were motivated by the concepts in quantum physics. So we treat each amino acid letter as a linear operator. So the linear operator works like this. So don't ask me why. I have tried many different kind of representation, and this one just work. So an operator applies to some coordinate x is simply defined as this coordinate plus the isometric embedding vector beta I. Beta I is this. It's just the vector we got from the multidimensional scaling. Left multiplied by this omega matrix. Let's leave omega matrix here. Just assume this is done. Okay. So take this short peptide sequence as example. So assuming that we want to write down the coordinate for this peptide, we begin from the rightmost letter A. So after this transformation, A becomes omega times beta A because there's no X. So this is simply 0. And then XEA equals to XA plus beta E left multiplied by omega. So we just plug this XA here. And for XTEA, you simply plug this XEA here. So that becomes, this is XEA plus beta T and left multiply omega. So you can write it on, and then you can get this, coordinate. So naturally, it satisfies non-commutability because when you switch the neighboring operators, you will cause a change in the coordinate. You can try that. And now we just leave the part, the isometry part to find the omega matrix. And that is actually also a challenging task. Actually, we spent a couple months in finding a good solution to this omega matrix. So I'm going to derive the properties of omega using several cases. The first one is a single mismatch case. So for example, you have two amino acid sequences. S1 equals AKAI, S2 equals to AKAJ. So they differ at the end, and the first letter is the same. So the coordinate of the first S1 is omega times beta I times beta K. And the X2 is written down as omega times beta J plus beta K. So you can calculate the Euclidean distance by x1 minus x2 square. So it's square root, but you can use square here. So it is simply written down as beta I minus beta J transpose times omega transpose square times omega square times beta I minus beta J. Because beta K term is canceled out, they have the same power of omega matrix. What is left is the omega square term. And you want this to be equal to beta I minus beta J transpose times beta I minus beta J, because this is the Blossom score. By definition of isometric embedding of Blossom 6-2 matrix, this is the Blossom score. So you want this one to become this one. And naturally, omega has to be a unitary matrix. So omega transpose times omega has to be the identity matrix. This is the first property. And the second, I just skipped the mathematical details, and with the two consecutive mismatch case, both letters are different. The desirable omega is like something like this. You increase the dimensionality by 2 times, and then you put the identity matrix on the anti-diagonal. So the omega 2 is an element in the second order cyclic group. The second order cyclic group is the most simple group in group theory that only consists of 2 elements, the identity element and another element G, a non-trivial element G. So satisfying G square equals to E. That is why it's called a cyclic group. So I'm introducing the concept of group theories not because I want to build a math wall so that people don't understand, but really it is helpful for us to derive the K consecutive mismatch case. So for 2 consecutive mismatch case, omega is an element in second order cyclic group. In the K consecutive mismatch case, it's simply the K's order cyclic group. So we in practice we choose K equals 6. Because 6 is actually good enough for CDR3 of length 17, even 14, 17, 16, 17, something like that. It's actually good enough. So it's an element in the 6th order cyclic group because this is the group. You have G squared, G to the 3rd, G to the 4th, G to the 5th, and then G to the 6th equals to the identity element. Okay, so this is the matrix we are looking at. And now, how about the performance? We compare the isometric distance with the Smith-Waterman alignment scores. But we first need to divide the CDR3s into different lengths because Gianna cannot handle, this algorithm cannot handle sequences with different lengths. We have to treat the sequences with different lengths separately. But you can clearly appreciate that I think for different lengths, it all aligns pretty well. The correlation is really high. The spanning of a certain scores at a certain score, the spanning of the isometric distance is becoming bigger and bigger, which is expected. But at the beginning part is very small. So actually, we only care about the low distance part because we are only focusing on the high similarity TCR clusters. So with that low distance, high similarity part, it performs actually very well. Okay, so okay, I have 15 minutes. So now let's talk about the Gianna workflow. We begin with TCR repertoire, and we perform sequential G6 encoding. We got the coordinates, and then we do nearest neighbor search. We got the pre-clusters. We call it pre-clusters because there's some noise in the original isometric embedding, as I shown before. So we still need to perform a last step of Smith-Waterman alignment to guard the accuracy of the final clusters, and that is how we get the final TCR clusters, and they're highly accurate. So compared to the state-of-art algorithms, Gianna can achieve 600 times speed up when compared to TCR dist. TCR dist is very accurate, so it's still used nowadays. And the iSmart is a little bit faster, but it's still very slow. And look at the accuracy. So pure cluster refraction can be sort of as understood as specificity. This is the sensitivity. You can see that Gianna, iSmart, and TCR dist, they're all similar, but Glyph is really much lower. So this is actually expected. Okay, so similar is true to sensitivity. So when another good thing about Gianna is that this mathematical framework allowed us to do very fast the query of new TCR6 samples against a large existing reference. So assuming that you have performed the TCR clustering of 10 million TCR sequences, and now you want to add one sample. Before, you have to add this one sample to the reference and then do the sequence clustering all over again. And this will take like several weeks to finish. But now you only need to calculate the isometric coordinate of the new sample, and then compare with the existing coordinates in the reference sample, and then just search for the nearest neighbors of these new TCR sequences, and then you are done. So that is very fast. So query against a reference data set with 20 million sequences only takes 3 minutes. So it's really fast. And it also allowed us to revise the concept of repertoire, revise the method of repertoire classification. So now we can simply apply Gianna query to do classification, which is a very important task in TCR repertoire analysis. So for example, we use a reference that has consists of 10 healthy controls and 10 COVID-19 patients. This is our reference. We know the labels of the T-cell receptors, whether they're coming from the COVID or they're coming from healthy individuals. And then we query of 100 COVID patients and 100 healthy control patients against this reference. We calculate for each of the query, we calculate how many of the TCRs are clustered with the COVID patients or clustered with healthy individuals. And if they are more often clustered with COVID, we will classify that a COVID patient. So with that, we have actually a predictor. So that predictor reached to 77% of accuracy for this very small reference. And when your reference grow bigger, your accuracy rapidly grow to near perfect. So for example, when you have 100 samples in the reference, your accuracy become 98%, which is as good as PCR test. So this is very encouraging. That is, so we apply this to study multiple disease classification. We include our reference samples. Our reference samples collected as cancer patients, a mixture of cancer patients, COVID-19 patients, multiple sclerosis, and healthy control. And we use unseen additional samples falling to the same disease category to do the query. And we find that with that simple query approach, the first line of diagnosis all achieved above 90% of accuracy. The first line diagnosis, I mean, disease versus control. And this is, so for the differential diagnosis task, it's actually much harder because they're all immunologically related, but still, I mean, the accuracy can reach to above 87%, suggesting that there is a huge amount of recurrent, I think, reproducible information in the TCR repertoire that can only be retrieved using ultra large scale TCR clustering analysis that Gianna is able to perform. Okay, so to summary, we have developed several tools in the field of TCR repertoire analysis, starting from calling TCR in the cancer, and then we go all along the way to do cancer diagnosis and cancer differential diagnosis from the other diseases. And of course, although we are on the track, we're still distance from our ultimate goal, which we hope eventually to achieve, that TCR-based early cancer detection. So with that, I'm thankful that I have very talented lab members, Dr. Hongyi Zhang and Daria, now has left the lab to a company, and Dr. Xuexin Yu and Jianfeng. And I'm supported by my close collaborators, Dr. Yang Xinfu, James Chen, and also my clinical collaborator, Jaya. She has generously donated a lot of clinical samples for us to develop the DeepCat algorithm. And throughout my career, I have received a lot of support from Kathy and Nir, the great couple that has been so helpful to my all my research efforts. My work is funded by NIH, NCI R1 grants and CPRIT and the Lida Hill Foundation. This is a lab gathering at 2019. We are not allowed to do gathering after COVID, but I'm ready to take questions. Thanks a lot for your attention.
Well, thank you very much for this really state-of-the-art talk. And I'm really looking forward to seeing how this progresses over the ensuing years.
Thank you.
Question, I'll take the opportunity to start the questions. And tumors have different mutational burdens. Does the TCR repertoire differ extensively between a very low mutational burden type of tumor and, say, a high mutational burden like melanoma or lung cancer?
That is a very good question. We don't have definitive answer yet, but we compared two cancer types very extensively. They are lung cancer and ovarian cancer. My understanding is that compared to lung cancer, ovarian is a much lower burden. And as you expected, lung cancer has a much stronger TCR signal compared to ovarian cancer. So it's a lot harder for us to diagnose ovarian cancer. But it is not impossible. It is still possible, but lung cancer has a much stronger signal even against COVID. So we can separate lung from COVID at high confidence, but not ovarian cancer.
Yes. Yeah. I mean, it was very exciting that you can identify, common tumor antigens such as that heat shock protein.
Yeah.
Do you expect there will be more?
Yes, we actually have a list. Some of them are keratin, some of them are associated with olfactory receptors. I can actually, we defined a list in our 2020 clinical cancer research paper that is reporting that method. But still, it's our ongoing research. We are hoping to find more with more advanced computational approaches.
Okay, so now Peter Van Galen from the Brigham and Women's asks, in the second part of your talk, are the neoantigens you described present in a large proportion of the patients, or do you foresee highly personalized targeted immunotherapy?
Okay, thank you for that question. The second part of the talk, we are mainly focusing on cancer-associated antigens. Because neoantigens are super rare, it is very hard for us to really get enough statistical power to compare across multiple individuals. So that being said, the percentage of patients that are carrying that cancer-associated antigen, for example, HSFX1, is about 15%. So I think it is possible there is some value to pursue with a follow-up, maybe a clinical study on that target.
And Anna Altamirano asks, this is really great for T-cell receptors. Can you look at B-cell receptors in autoimmune related diseases?
Yes, so that is absolutely possible. We really wanted to. So for this many years, I have always wanted to study the B-cell receptors as well. But there are not as many B-cell receptor sequence data in the public domain as TCR. So as we're speaking for now, the TCR data is about 10 times more than the BCR sequences. So we are still waiting for more data to be generated.
And Matthew Bernstein asks, regarding the DeepCat algorithm, is it possible to extract sequence features that the algorithm is using to make its decisions? And if so, are there any biological or clinical insights to be gained from these features?
That is a very good question. So it touched the very core of the black box of the deep learning. So we know that something is going on, but we don't know what it is. Unfortunately, so far we have done some sort of reverse engineering of the deep neural network model, but we couldn't find a very clear explanation to what it is. Some of my wild guesses is that there are some biased usage of certain amino acid biochemical properties that is being picked up by the algorithm. But why is that? What is the cause of that? It is currently unclear.
And Yilin Key asks, the tumor-specific TCRs in the peripheral blood seem to increase with the stage of tumor progression. Have you compared whether there are any correlation between the frequency of tumor-specific TCR with the response to checkpoint immunotherapy?
First, thank you for picking up that. I forgot to mention that yes, the cancer score increases with advanced tumor stage. I have made many attempts trying to figure out if the signal is predictive of immunotherapy responses, but so far it's not successful. I have tried DeepCat, I have tried iSmart, I have also tried Gianna. Unfortunately, none of them is very successful. So we're probably waiting for more data to really generalize a lot more. Yeah, thanks.
I know Science Magazine's breakthrough of the year was computer prediction of structural interactions from sequence. And what do you think are the necessary steps, Asaf Mosius asks, towards solving the problem of predicting TCR antigen specificity from the TCR sequence?
That is exactly what we're thinking right now. So AlphaFold and AlphaFold 2 and the Rosetta Fold, both of them, they can only handle a single channel. Okay, Rosetta Fold 2 is able to handle a little bit of protein-protein interaction. But for the specific problem of T-cell receptors, the biggest challenge is that you cannot find homologs. Without homologs, none of them will work. So I think the biggest challenge here is to specifically model the T-cell receptor interaction with the MHC peptide complex. And that is, I think, I don't know if there's enough data in the public domain for us to perform that prediction. Yeah.
Okay. And Yi Zhang asks, nice talk, Bo. Given that the blood TCR-based cancer score may also link to autoimmune disease, what is your thought about cancer detection using other measurements from blood? For example, the concentration of certain cytokines compared to TCR measurements.
This is absolutely a great question. So I think a lot of people in the liquid biopsy field are thinking about the possibility of using multiple bioanalytes in the blood to achieve the higher sensitivity and specificity at early stage. I think the advantage of TCR-based detection is high sensitivity, but low specificity because of the other immunological conditions. And I'm hoping that adding the other components in the blood will actually achieve a better specificity. So that is, yeah, you're right. I think a combination will be a future direction.
Yes, I wonder whether looking simultaneously at B cell receptor and T cell receptor diversity might be different between cancer and autoimmunity, or have any differing signals.
I think that's absolutely an orthogonal signal to add, because B cell and T cell act on different stage of immune responses. So if we can and have enough data, that is absolutely my immediate next step. But as I mentioned before, I mean, for now, there's a very limited data set that we could develop for a machine learning algorithm. And I'm really reluctant to do that, without enough data. But really, this is a great new direction that we can, I think about a lot of new opportunities.
What is more difficult or different about the reason that more B cell receptors aren't in the databases? Is there anything we need to change the sequencing
that is
strategy?
I think no, actually, I think they're just for people who are studying T cells, TCR repertoire analysis is a very natural tool to really describe the population behavior and to link that to some of the disease properties. But for the BCR, I think for antibody people, this is not their first go-to kind of tool. I mean, they have many other kind of techniques, like clonal, isotype switch and somatic hypermutations. I think they have other ways to detect which of the B cell antibodies are of high interest. But that being said, in recent years, I see some very exciting trend that people are also applying high throughput BCR sequencing technologies. So I'm expecting that in the next 2 years, this will become very available.
And Tommy Tang asks, a great talk, thanks. What's the cancer score cutoff that should be used? Or is it cancer type specific?
In the Science Translational Medicine paper, we reported a universal cutoff for all the cancer types. There's no way that we can distinguish different cancer locations right now. The cutoff is based on the training data. It's about, I think, 0.24, something like that. But it's really preliminary. So before any further prospective clinical study, we cannot set that for stone.
And Yoki asked, how applicable is this to hematologic malignancies? For example, hematologic cancers that get misdiagnosed in earlier years as other subtypes, such as blastic PDC neoplasms. And I think I would also add, Hodgkin's lymphoma has a very high response to PD-1 immunotherapy, but is not considered to be highly mutated. Is there what's the TCR repertoire in Hodgkin lymphoma?
I'm very happy to say that we have actually applied our method on B-cell Hodgkin lymphoma samples published by actually Margaret Shipp about 2 years ago. So that actually works pretty well. We were able to separate the healthy donors from the lymphoma patients at very high confidence. So I guess there's still some so that cancer signal may not entirely depend on the somatic mutations. There's some mysterious kind of immunological response that is going on in the repertoire that's causing the systemic change. We're still trying to figure out. Sorry, I forgot what the second question.
It was, yeah.
I think it's related to PDC. My apologies for that, but we currently cannot really separate different subtype of blood cancer types. We don't have enough data to really try that, but that is an interesting way of applying our method.
Mm-hmm. And Stan Gu asks, very exciting work, Boo. Did you detect HLA subtype associated TCR features?
No, but thanks, Stan, for the question. This is asked by reviewers as well. People kind of have have a difficult time understanding why patients with very diversified genetic background could all have very similar immune response. I think the short answer is that it is not. It's probably because that different HLA allele could present some shared antigens like HLA2 and HLA3. If you see the prediction, I think a certain percentage of the antigens, both of them can combine to, but that's not the ultimate answer. The ultimate answer, I think, lies into T cell differentiation. I think cancer patients may have a digressed or abnormal way to steer the T cell differentiation from the very beginning. So there's some systematic huge difference in the repertoire. Yeah.
Well, I'd like to thank you for your talk. I learned a lot and really look forward to hearing more in the coming years about how this gets applied.
My pleasure. Thank you so much, Gordon, for great hosting. And thank you everyone for listening to my talk. This is my great pleasure today. Thank you.
Thank you.